First up this morning will be the Biosystems Division, talking about their work with microbe-host environment interactions. And I'd like to introduce Professor Ina Hoeschele here, and she'll be starting us off. Are we good?
Yeah, thank you. Good morning. So my background in research area is statistical and quantitative genetics. Statistical genetics is just a branch of biostatistics. And it obviously strongly overlaps or encompasses quantitative, population, evolutionary genetics, and it can be viewed as statistical genomics and bioinformatics with the added value of genetics. A particular area of interest at the present time for statistical geneticists is systems genetics or genetical systems biology, which is a field that essentially merges the problems and methods from genetics, systems biology, and genomics, and has been described as the next frontier in systems biology and medicine. The current main focus of my research is on developing and applying statistical methodology for the interpretation of large-scale multi-omics human datasets. So here we have a diagram of the influence of genetic variation or DNA sequence variation, which of course influences gene expression or mRNA levels, subsequently protein levels and structure. Proteins function together to determine events at the microphysiological level, flow of metabolites within and across biochemical pathways, which then in turn dictate events at a more macrophysiological level, which then influences subclinical phenotypes, which eventually lead to clinical phenotypes. So as an example, we have cardiovascular disease, and subclinical phenotypes for cardiovascular disease are measures of atherosclerosis or plaque accumulation in the arteries, which can be measured using ultrasound imaging techniques. And the influence is not just from top to bottom, but we now know that environmental factors and risk factors, more generally risk factors, for instance, body mass index, right, is a risk factor for CVD, cardiovascular disease, that these risk factors influence mRNA levels through epigenetics, for example, DNA methylation. And in the past, essentially all we have done in these so-called genome-wide association studies has been to associate DNA sequence variation with the clinical phenotypes. And these studies require national and international consortia and thousands to 10,000s of individuals, so they are expensive and difficult to organize. And although they certainly are continuing to make important contributions, today we associate DNA sequence variation with all levels of this hierarchy. So they are expression SNPs, they are protein SNPs, they are methylation SNPs, they are SNPs influencing the subclinical phenotypes, they are SNPs influencing risk factors such as BMI, and typically the effects of these SNPs on all of these intermediate phenotypes are much larger and much easier to detect than the effects on clinical phenotypes. So I consider myself very fortunate to be the lead statistical collaborator for several multi-omics projects that are based on the multi-ethnic study of atherosclerosis, or MESA. MESA is a prospective cohort with almost 7,000 participants from 4 races or ethnicities. And the phenotypes measured in MESA are all related to atherosclerosis, so mostly different measures of plaque accumulation. Of course, there's also a collection of extensive clinical, social demographic, lifestyle, medication, nutrition, whatever data. And these data have been collected at baseline in 2000, I believe, and at several subsequent exams. Exam 6 is planned for next year. I forgot to mention that MESA, my collaborators are at Wake Forest Medical School in Winston-Salem, which is one of the founding institutions of MESA. And in MESA, genome-wide SNP data are long available. And the data that we have currently added to MESA at one particular exam, exam 5, genome-wide data on now approaching 2,000 individuals, including gene expression, DNA methylation, and microRNA at the present time. Of course, some limited proteomics and some other things. So this study and this comprehensive data set allows us to really study this hierarchy. So what we have done initially, of course, is to associate gene expression with the atherosclerosis phenotypes, to associate DNA methylation with the atherosclerosis phenotypes, to associate, of course, in detail, to investigate the association between methylation and gene expression, to associate risk factors with methylation and gene expression, to perform mediation analyses of the effect of DNA methylation, which is mediated by gene expression on the subclinical phenotypes, to perform mediation analysis of the effect of risk factors, which is mediated through methylation and gene expression on the subclinical phenotypes, to associate DNA sequence variation with expression, with methylation, with subclinical phenotypes, with risk factors. And as I mentioned before, we have done this at one particular exam, so this has been essentially a cross-sectional study. We are now expanding the study into a longitudinal study, pending funding, because MESA has data at different exams and continues to plan future exams. And we will also focus more going from association and mediation analyses to prediction of disease. So one particular interest that I currently have is to use the DNA sequence variation to perform causal analyses in this data set. So randomization is very well known to be the most convincing method for establishing a causal relationship between any two variables. And Mendelian randomization uses randomization of alleles during meiosis, which of course precedes the effect on a molecular or clinical phenotype, gene expression, atherosclerosis. So Mendelian randomization uses the randomization of alleles during meiosis to perform causality tests that are less susceptible to hidden confounding. MESA is an observational study, so confounding is of course a big issue. Much of the early Mendelian randomization work has focused on estimating the effect of a modifiable risk factor such as BMI on disease outcome, CVD, using a single SNP. My interest is on network, something that I call network Mendelian randomization, which uses multiple SNPs, other external variables, essentially to compare different, to not perform mediation analysis conditional on a certain causal model, but to compare different causal models using structural equation modeling. So in particular, we are interested in our data to identify those genes for which this causal chain holds, right? We have a risk factor such as BMI that influences, leads to changes in DNA methylation, which leads to changes in gene expression, which leads to changes in atherosclerosis. And to identify such a causal chain is not trivial because, first of all, there is the potential for hidden confounding for any of these links. There may be a confounder between methylation and gene expression or gene expression and atherosclerosis. And there's the potential for reverse causation for any of these links. Gene expression can be responding to a change in the subclinical phenotype. And of course, there are cases where gene expression actually regulates methylation instead of the other way around. So we think that this type of Mendelian randomization analysis, causal analysis, could be which we are currently exploring in detail, could be an important step between these types of genome-wide omics association studies and clinical trials. Thank you.